So welcome everybody. Tonight we have Dr. Nick Mason presenting. He is an assistant professor in the Department of Biological Sciences and is also Curator of Birds at the Louisiana State University Museum of Natural Science. He's originally from Minnesota. Nick did his undergrad at Vassar College, his master's at San Diego State University, his PhD at Cornell University, and a postdoc at UC Berkeley and the Museum of Vertebrate Zoology before joining LSU in the fall of 2020. Nick and his lab group at LSU have wide research interest in ornithology, including systematics, biogeography, song, color, and understanding human impacts on bird populations. Natural history collections provide a unifying theme throughout their work, and the Mason Lab is dedicated to documenting and studying biodiversity through ornithological collections, both locally and abroad. Outside of ornithology, Nick enjoys gardening, painting, and playing rugby. And with that, I'm going to turn my mic off and pass it over to you.
Great. Thank you so much, Katie, for the introduction, and thank you everyone for tuning in. It's too bad that we can't be together in person. Hopefully things will improve with Omicron and we can meet in person in the future. But regardless, I'm excited to have an opportunity to present sort of my idea of the LSU Museum of Natural Science to all of you. And I think it's really important that we have a connection between local interest groups like Audubon Society and academic units like LSU and the museum on campus. I think we have definitely some shared mutual interests, and I think fostering these connections is really important. So I'm excited to share with you sort of some of my work that my lab group does. And I've also included kind of a retrospect, a little bit of a history on the museum, just some of the people and time points that were important in terms of the development of the museum. I've had a lot of fun moving here. This has been a dream job ever since I got involved with ornithology. I've known about LSU as a powerhouse and a really well-respected place for ornithology, and also just really strong community. And so I'm excited to share some of this with you tonight. So it's kind of interesting when you think about what exactly is a museum, and this means different things to different people. But I wanted to start here in thinking about how we think about museums in sort of general society. So we can think about it as a collection of interesting things, and this was the first image that came up when I did a Google image search for museum, maybe a place for contemplation, or possibly a way that we can try to preserve items of cultural importance. And not even that too long ago, at least certainly in evolutionary time, museums were sort of preserved for really only those who were deemed worthy in a way that possessed the qualities prerequisite to appreciating music, philosophy, art, literature, and science. So who benefits from a museum and who has access? And this is sort of an idea I'll come back to periodically throughout the talk. But I wanted to start off by thinking about what the LSU Museum of Natural Science does. And there's 5 main things that we do. The largest and most prominent thing that we do is we generate new knowledge. And so we're a community of scientists, of naturalists, and of conservation biologists that are geared towards trying to understand the natural world. And we go out and we collect data from the field and from the lab and from theory, and then we apply that to try to understand nature. We also try to collect and preserve specimens as a record of Earth's history. And this is often counterintuitive to folks at an initial glance. They may think that museum specimens are something of the past, sort of an anachronism of the times of Audubon. But we believe wholeheartedly here at the Museum of Natural Science that continued scientific collecting is really important and is essential really to understand how our planet is changing and how those changes are impacting bird populations. We also educate and train the next generation of scientists. So here are a couple of our recent graduates, Oscar Johnson and Jesse Salter, who recently finished their PhDs and went on to do postdocs at different institutions. And this is a big part of what we do, training undergraduate and graduate students to become the next generation of scientists. And like I'm doing tonight, and hopefully we can continue to do, I think it's really important that the museum engages in outreach. And part of what we've done that historically is through the public side of the exhibits that we have at the museum, but I think also it's important for us to engage with local naturalist groups like we're doing tonight and encourage stewardship and education of the general public. And finally, one other kind of interesting thing that we do is we often are sort of a local reference or authority in terms of identification and consultation services. So just as an example, one of my graduate students recently helped out a government agency in terms of identifying some illegally procured specimens that were taxidermied. And so we also sometimes help out locally, but also federally in terms of identification and consultation. So that's sort of the biggest lens what the museum does. But today what I really want to do is talk a little bit about the history of the collection. And for those of you who may know the collection well, some of this may be old hat or old news, but I've been really enjoying learning more and more about the people and the history involved in this collection. It's a really fascinating story. I'll talk a little bit about ongoing research at the museum and showcase some of the ongoing student work. I'll talk a little bit about my lab specifically and the kind of work that we do, and then a little bit just on sort of my vision for the future of the museum and hopefully how we can interact with Audubon and other parties into the future. So I want to start with just a brief history, and I'm going to try to keep this relatively short. A lot of this focuses on the people that were important in the formation of the museum and sort of the earlier stages. So the museum was founded in 1936, and if you've been in the museum, hopefully you have. If you haven't, hopefully you can come visit once Omicron settles down and we can reopen to the public. But there's a research side and there's a public side. And so the research side was founded in 1936 by this gentleman, Dr. George Lowry. He was an undergraduate at LSU, was born in Monroe, Louisiana. And he became a pioneer in developing this technique to observe migratory birds by watching the moon and actually counting shadows of birds or silhouettes as birds as they went across the moon, and also wrote some really authoritative natural history books on the state, including Louisiana Birds and Mammals of Louisiana. And there's this really excellent document called Windows of the World, and I'm actually going to put this in the chat in case anyone is interested. But if you're curious to learn more about the history of the museum, I just put a PDF there in the chat. That's a link to our website at LSU, and it has this really interesting history of the museum, and I encourage anyone interested to read it. But one of the things that George Lowry really believed in was that the entire museum, including both the research collections and the public exhibits, are a really important jewel on LSU's crown. And so for a long part of George Lowry's career, he really fought to make those public exhibits an important part of the museum. And we at the museum are also encouraging outreach. And it's really interesting to think about how the space in the museum is dedicated to different components, whether it's research, whether it's outreach. And that's something I'm going to come back to because that's something that's actually changing in the museum and in the building. But George Lowry unfortunately passed away suddenly from a heart attack in 1974. And at that time, John O'Neill became the director. And so George Lowry was the director of the LSU Museum from when he founded it until his death, all essentially 40 years of service to the museum. He really saw it through. It was his life's mission to create this museum and to build it. And he did an amazing job, and we're really indebted to him. So John O'Neill took over the reins in 1978. John O'Neill was also a fantastic painter. Here's one of his paintings. And if you come visit the museum, you'll see he has a bunch of incredible paintings. He has also described various new species to science. This is one of them. This is the orange-throated tanager that was described by George Lowry and John O'Neill in 1964. And around this time is when LSU really started to establish itself as a community that was focused on the neotropics. And so during this time, a lot of really prominent ornithologists came through LSU, and one of the most famous of which was Ted Parker. So if you haven't heard of Ted Parker, he is a fascinating character who unfortunately passed away at an early age in a plane crash. And so often it is romanticized, but it's incredibly difficult. People give their lives to the study of birds. And Ted Parker is one of those people. He was an amazing ornithologist. Some describe him as the most gifted field ornithologist of the 20th century. He was a research associate at LSU, but really he's much more than that. He's sort of an inspiration to a lot of us at LSU for thinking about how we can observe and learn more about the natural world. And I'm going to put another link in here because Ted Parker is a really fascinating person. Upon his death, there was a whole issue in Ornithological Monographs that was dedicated to him. The first 2 chapters are a short biography written by Van Remsen. And if you want to learn more about Ted Parker, I really encourage it. He was a fascinating person and a really amazing naturalist. So he's definitely sort of an honored figure in the history of the museum. And he actually plays a prominent role in this book, if you haven't read it, that also deals with a lot of expeditions during this time. It's called A Parrot Without a Name, and it's actually a really great read. So if you're interested, I encourage you to check that out too. So shortly after George Lowry's death, Van took on, and so I'm guessing that many of you know Van Remsen. He's a very prominent figure in Louisiana ornithology. He's an expert naturalist. He's now a renowned expert on Louisiana birds, but also is an incredibly knowledgeable person regarding South American birds, or just birds worldwide. He has an inexhaustible knowledge of birds. He established what's called the South American Classification Committee, which makes a lot of important decisions in terms of taxonomy, and is a renowned authority on Louisiana birds. During this time, shortly after Van Remsen was hired, then Bob Zink was hired for a couple of years, and this is sort of the beginning of when LSU and the museum started focusing on what's called molecular systematics. And that's still a really primary focus of our museum. That's where we use DNA to understand how species and populations are related to each other and try to reconstruct their evolutionary history. So Bob Zink played a really prominent early role in establishing the genetic resource collection that we have here, and it's now the largest collection of genetic resources in the world. So Bob Zink was here for about 9 years, and in doing so, he played a really important role in sort of establishing one of the main directions of the museum that continues to this day. Around this time also, Donna Dittman and Steve Cardiff were hired, and they've been figures at LSU for a long time now, working at the museum for over 30 years, and they've trained countless students and collected tons of specimens to help the collection grow. And they've also become leaders within the Louisiana Orthological Society. I know a couple of other folks in the audience went to the Rice and Rails Festival, and that's a fantastic birding event. If you haven't been to it, I highly encourage you to check it out. Fred Sheldon was hired in 1994 and is actually retiring this semester, so we're going to have a symposium celebrating him later. But he was, again, another pioneer in early stages of avian systematics, biogeography, and molecular evolution. And he really is focused more on the geographic scope of outside of the neotropics, with a lot of the previous folks at LSU have been working on, to expand into Southeast Asia, Indonesia, and other regions of the Malay Archipelago. And then last but not least, Rob Brumfield joined in 2003 and is, again, an expert on speciation and systematics, and specifically on subaquine birds. They recently published a comprehensive phylogeny of this group. It's a clade, or a group of about 1,000 species. And so this was decades in the making of fastidious, really hard work in the neotropics. He's now an associate dean in the College of Sciences at LSU. And so collectively, we have this whole community of ornithologists here at the museum, and really I'd like to emphasize that part of it. And we're really proud not only of the work that we do, but the people that have come through LSU. And so this is just a sampling of the folks that have come through and now hold various positions at different museums throughout the country and throughout the world too. And so that's one of the things that we're really dedicated to, is trying to train folks in understanding curatorial sciences and how we can use museums to understand ornithology. I also just want to give a shout out that there's a very active ornithological community at LSU beyond the museum too. So Phil Stouffer, I know a couple of folks in the audience have worked with him, Sabrina Taylor, Ashley Long, Kevin Ringelman, Mike Polito, Jessica Eberhard. So there's tons of ornithologists here at LSU, and it's really a great community, and it's something I think we can celebrate as a group. And also, it's not just for the birds too. So there's also really fantastic curators in other groups in anthropology, ichthyology, geology and palynology, mammalogy and herpetology as well. And so I feel like, I have a dream job, and I feel privileged to be part of this group and to work on birds for my career. And I just encourage you all to try to engage and interact with us, because we really are sort of a special group in a special place, focused on museums as a way to try to understand the natural world. So a little bit more just on the bird collection. So this actually is one of the most active collections in the world, especially since 1980. We have We just did our tally. We have more than 220,000 bird specimens total, and it's the third largest university-based collection in the world. So you can see in this plot here, since it was founded in 1936, this is an accumulation of study skins. So you can see it's been a pretty sharp increase since 1980, and this is continuing. And so we've also started to diversify the types of specimens that we collect. And so we've started to collect more of what are called pickles or whole alcohol specimens. And with those, you can actually get information on the internal anatomy of the bird. And so when we prepare a study skin, a lot of the internal organs and skeletons are actually discarded. And so we actually lose some information there. And so one of the things that we've been doing recently is trying to diversify what types of specimens we collect when we go out and collect specimens. And so we recently updated our data. There's a portal that you can explore all the museums and all the collections that are across the world. And this is the global distribution of bird specimens that we have at LSU. And so colors that are more red are higher numbers. And so you can see we have a really high concentration of specimens here in South America, but also, as I mentioned, so Fred Sheldon and other folks have been focusing on Southeast Asia, but not so much in Africa. And we do have some for the southeastern United States and also for the western US. And so this gives you a sort of a snapshot idea of where we focus geographically in terms of the history of the museum. And Peru and Bolivia specifically has been the central focus of the Museum of Natural Science here at LSU. We've been working there almost every year since the 1960s. And much of the collecting, so this is a sort of a heat map again showing where most of the specimens have been collected. So most of them have been collected in lowland Amazonian areas. And I'll talk about this a little bit more, but one of the things that I've become interested in and hoping to focus on is actually working more on some of these high elevation areas in the high Andes. But you can see we have lots of specimens from the lowland Amazonian areas. And so folks like Rob and Van have focused a lot on understanding speciation and diversification in Amazonia and the role of Amazonian rivers and things like that in contributing to avian biodiversity. One of the other things we're really proud about in the museum is that our dedication to field work and international field work in some really remote areas has led to the description of a lot of new species. And so there's been 46 species that have been new to science that were described by LSU personnel since 1964. Here are a couple of my favorites that I've just selected from them. This is Xenoglax lowrii, the long-whiskered owlet, and this is black-faced cotinga, Inca wren on the top left. So some amazing new species that have been described based on the hard work and dedication of a lot of our field biologists. And this is continuing to just recently. So just last year, actually, we had another new species that was described by Dan Lane. He's an LSU Museum of Natural Science Research Associate. He is one of the best birders in the world, I would say. He's a tour guide and a fantastic birder. And so he and some of the other LSU associates here described this new species, this bright yellow tanager from northwestern Bolivia and southern Peru. They called it the Intitanger, which is the Quechua or the indigenous language. It's the word meaning sun, so sun tanager, and it's named after John O'Neill. And it's thought that this, despite being this bright yellow bird, it's been hidden to science because of its migratory patterns. And so it's thought that it breeds during the rainy season in this deciduous forest, but then during the dry season, it actually goes to this purple area and sort of a little bit lower elevation. So it's a migratory bird, but it migrates within the tropics. And it has a fairly localized endemic range to this very specific part of the Andes Mountains. So we're really proud of this aspect of the museum-based fieldwork that we do. And this is just another example that there's still things out there to discover. Okay, so I want to move on a little bit now and talk about some of the work that's ongoing at LSU. And in general, I think I've probably tried to pack too much in this talk. I apologize for that, but I'm going to try to showcase some of the different work. And my hope in doing so is that you get a sort of a brief glimpse of the breadth of work and the people that are involved at the museum. So there may be a lot to take in, but if you have any questions on any of the specific projects, I'd be happy to answer them. But basically, I asked grad students to give me a slide summarizing their either ongoing or future research interests. And so I'm going to go through those here to give you a sense of the kind of work that folks are actively doing in the museum. And so first, this is Diego Cueva. Diego was from Colombia originally and is interested in this Manicus mannequin systems. This is actually a group that Rob Brumfield worked on in the past. And so Diego's interested in speciation and hybridization. And so there's these two hybrid zones with this purple arrow that are shown here, where the white-bearded and yellow-collared mannequin come into contact. And so hybrid zones are these really fascinating natural laboratories that we can use to try to understand how reproductive isolation evolves. Each one is different in its own dynamics, and that can relate to the life history, ecology, geology of the organisms. And so there's really fascinating sort of particle colliders that exist in nature that we can use to try to understand evolution and ecology in action. Next, we have Eamon Corbett. So Eamon Corbett actually gave a really excellent talk to Louisiana Orthological Society on this system, and I think that's on their website if you want to check it out. But he is interested in, again, hybridization, as well as eye color and the genetics behind eye color in gray-tailed and boat-tailed grackles. And so he has been studying and sampling specimens or populations from different parts of the coast. And here in Louisiana, we have a putative contact zone between these types. And so again, it's an interesting system for trying to understand dynamics of gene flow and how signals like eye color may be involved. So far, he hasn't found any evidence of hybridization, but he's continuing to work on this. And yeah, so stay tuned. Roberta is actually in Brant Faircloth's lab, but she's interested in structural color. And so she's looking at these Lepidopteryx mannequins. And so you can see there's variation in plumage on the bottom left here, actually. So you can see these are different subspecies, or maybe species too, I believe, but they have differences in the color of their crown. And so she's interested in both the color and the reflectance of that, but also using electron microscopy to get at the microstructures that are involved in generating these colors. And so structural colors, how they evolve, and how they might be related to gene flow. And in the same system, Andrea Moncrief is a more senior grad student in Rob Brumfield's lab. He's been working on the blue crown mannequin system, which is the same group, but more thinking about the phylogenetic history of this group. And so you can see that there's these different subspecies or lineages within this species complex that are distributed throughout the Amazonian basin, as well as into Panama and Central America. And he's found some pretty distinctive clades or groups within this that probably are separate species in what is currently considered a single species. But he's interested in, again, this process of population genetics and phylogeography and these trying to understand how genetic variation is partitioned among populations and how that can reflect the evolutionary history of a group. Marco Rego, you can see him down here. He's a Brazilian, and he makes these, let's see if this works. Oh yeah, there it goes. So he has these fantastic maps that illustrate the distribution of museum specimen records. And so he's interested in trying to understand how complete are our collections in trying to be representative of different populations and different species. And so he's a wizard with using GIS and creating maps. And using this, he's actually able to find where there's turnover, where there's changes in bird communities. And so basically in this map here of the Amazonian basin, these bright yellow colors indicate where there's turnover, and not coincidentally, they correspond to the major rivers of the Amazonian basin. And so with this data, he's able to characterize and quantify how rivers are important in terms of dividing lineages of birds. So Marco's partner is Glaucia Del Rio, and let's see, there she is. Yeah, so Glaucia works on a hybrid zone, specifically these Rigmatarina antbirds. And so there's these two antbird species that hybridize in Amazonia. And she's interested in trying to understand why these two don't just collapse and become a single species. And to do this, she uses a lot of different tools. She looks at incompatibilities between mitochondrial DNA and genomic DNA. And so when we breathe and we create energy, there's an interaction between mitochondria and nuclear cells, and basically that interaction can sometimes break down. And so she's interested in trying to understand the dynamics and the genetic components of that, and found that there's actually a chromosomal inversion between these two groups. So there's part of a chromosome that was flipped, and that is actually dictating some of the hybrid zone dynamics between these two species. And she just she just accepted a new position at Cornell, and she'll be going there for her postdoc. Oscar Johnson recently defended his PhD, and he worked with Rob Brumfield on the macroevolution of subocean birds. He has also now gone to a postdoc with another LSU alum, Mike Harvey, and he's interested in understanding how long it takes for speciation to occur. And so he combines a bunch of different data sets, including morphometrics, plumage, song, and a phylogeny, to try to analyze macroevolutionary patterns and sort of broad scale trends in terms of how different traits evolve and how that might relate to when lineages diverge and become two different species. Another recent graduate was Rafael Marcondes, and he also worked with Rob Brumfield on the variable antrike complex. And so this is one of the most variable species in South America. There's tons of different subspecies that have sort of minute variation in coloration and patterning. And he found that there's this agouti signaling protein gene, which has become more and more prominent in a bunch of different systems. And so in this same gene is responsible for a light versus a dark coat in mice, as it is versus light versus dark plumage in these antbirds. So Rafael is still actually here at LSU and is now an instructor. Okay, so that was kind of a whirlwind, but hopefully it gives you a little bit of a sense of some of the different projects that are going on at the museum. I'm going to shift gears a little bit and talk specifically about my lab group. And so I just started here in July of 2020. And so I have a small but growing lab group that consists of 2 graduate students, David Vanderplum, Samantha Rutledge, and then 2 postdocs, Subir Shakya and Maggie McPherson. And so I'm going to briefly talk about their interests and then transition to kind of projects that I've worked on in the past and things that I'm going to be doing while I'm here at LSU. So Samantha just started, and she's got a lot of different interests, as you'll see, but she's generally interested in thinking about structural color and how that might relate to hybridization, and also how human impacts are changing dynamics of those hybrid zones, and also how we can try to use genetics and genomics to try to improve conservation efforts. And so she's at the phase where she is thinking about a lot of different ideas, and we're actively trying to narrow that down towards a dissertation project. In the past, she did some really awesome work looking at brood parasitism and the dynamics of how birds actually perceive different colors of eggs. So yeah, that's Sam. And then next we have David Vanderplum. So David is a second-year PhD student, and David's interested in altitudinal migration. And so many birds will undertake long-distance longitudinal migration, but less well-studied is that many birds will actually shift their distribution elevationally across seasons. So David's interested in studying this phenomenon and how that might relate to divergence between populations. And specifically, he's going to be focusing on the slate-throated redstart, which is a warbler that's pretty widespread from Mexico all the way down to Bolivia. And it has a lot of geographic variation in color, but also in its migratory behavior. So he's interested in how that might contribute to diversification in the species. Maggie McPherson is a postdoc in my lab group, and she's interested in how biogeography and migratory behavior shapes diversification in birds. And she uses museum collections, as shown here, to get a sense of morphology and how that might relate to migratory behavior. She's also done a lot of really cool work related to behavior, specifically in great-tailed grackles, where she's looked at sort of novel problem-solving behavior in different groups. And she's interested in kind of spatial ecology and trying to understand how organisms respond to changing conditions, both cognitively, but also in the sense of across seasons, and how they maybe might change their migratory behavior or their distributions in response to global climate change. And then this last slide is for Subir Shakya. So Subir is here in this image prepping specimens in Southeast Asia. He's a really accomplished field biologist, and it might be a little bit hard to see this, but he's kind of shown a little bit of his map and all of the different things that he's gotten to enjoy, including gators here in Louisiana. These are some of the species he's worked on. He also recently described a new species a couple years ago, the cream-eyed bulbul. And so he is a biogeographer interested in Southeast Asia, in the diversification of Southeast Asian birds, but also interested in the genetics of color and maybe the genetic underpinnings of variation in color among populations and species. So if we step back a little bit, my lab group and myself were interested in these two kind of main topics, thinking about the generation of biodiversity that is with respect to phenotypes, and so the appearances of different birds. Why do so many birds look so different from each other? And how are there so many species of birds? And why are different lineages variable in terms of the number of species that they contain? So that's what we're interested in terms of the generation of the biodiversity. But we're also interested in how that biodiversity can be maintained. The planet is changing really rapidly. Things like agriculture or climate change, sea level rise here in Louisiana are changing habitats. And so how can we try to maintain the biodiversity that we have in the face of global change? All right, so I'm going to shift gears a little bit again here and just present some more vignettes. So this is, I realize that there's a lot going on, but I'm going to basically show some examples of these different foci of my research group, drawing on some of the work that I've done in the past or that are ongoing projects. And so one of the main things that I'm interested in is understanding speciation and what's called systematics in birds, basically understanding the evolutionary relationships among birds, as well as taxonomy, and so our classification of birds. And so I'm going to present two ongoing and recent examples from this work. So one group that I've been working on are the redpolls, the redpoll finches. These are eruptive finches that I think they made it down to Louisiana a couple of times. I was looking to see if there were some records. I'm not sure. Maybe someone can verify that for me. But they are eruptive, and so during some years they will come south in search of food. But there's these three recognized species, and many hardcore birders will spend a lot of time in the cold trying to identify a hoary redpoll on the left. And some of the work that we've done has looked at genetics of this group. And down here in the bottom left, this is something that's called a structure plot, and this is a way that we can visualize genetic similarity. And essentially the fact that all of these bars look the same, and that they have representatives of these three putatively different species, suggests that they're all extremely closely related to each other. And so we actually think that there's probably just a single species of redpoll worldwide. And so we proposed this change back in 2015 on an initial study that was turned down, but we have a new paper that came out just earlier in December, where we show that they're still extremely similar, and there's just a few very small genomic regions that are actually responsible for the differences in appearance between these species. And so birders might not like the idea of losing a tick off their species list, off their life list, but we think that this adds more to the nuance of our understanding of the biology of the system. It's kind of neat to think that basically just a few changes in the genetic composition of these individuals can have such a big impact on their appearance that we perceive them as different species. So this was work that I did with Eric Funk and Scott Taylor, who are both at UC Boulder. Oh, thank you, Cled. I appreciate that. So the next one I wanted to talk about may make birders a bit more happy, a bit more excited, is the Stellar's jay. So this is a project I did as a postdoc at the Museum of Vertebrate Zoology, and it's ongoing. So we've just finished our first draft. We're about to submit it. So this is hot off the presses, but essentially what we find is that Stellar's jays have this really deep genetic split between them. And so if you can see that, that's about 8% in mitochondrial DNA. For frame of reference, humans and chimpanzees are about 99% similar in their DNA, right? So this is a pretty deep divergent split between these populations of Stellar's jay. They also seem to differ in terms of their phenotype and different aspects of their ecology. And there's this really narrow hybrid zone here in northern Utah, southern Idaho, where they do come into contact and they do hybridize, but pretty infrequently. And so we think that these are reproductively isolated lineages of Stellar's jays. And so you can hopefully look forward to having another species of Stellar's jay, where we would have one sort of a Pacific Stellar's jay, and then one in the Rocky Mountains that goes down to Mexico. So some of what I do and some of what my lab group does is to try to use DNA as a tool to understand the evolutionary history of this group, or of birds, I should say. And often that has implications for what we classify as a species, whether it's lumping multiple species into a single species or whether it's splitting one species into two, but trying to improve our classification, which reflects understanding of the evolutionary history of these different groups of birds. Okay, so I'll briefly talk about a couple of different, or one project, I guess, in biogeography. But biogeography is this idea of layering geography on top of a phylogeny to try to understand the history of lineages with respect to changes in landscape or dispersal. So for this, I'm going to discuss a group of birds that are called the white-collared seed-eaters. And so this is a group of birds in the genus Spirophila. And when I started this project, there were 3 subspecies of birds that were recognized within this group. These are what are called tanagers from the family Thraupidae, true tanagers. So our Paranga tanagers that we have, like the scarlet tanager and summer tanager, are actually cardinals. These ones are in the family Thraupidae, so the Central and South American neotropical tanagers. And you can see their distribution here. And so there's one subspecies on the Pacific side of Mexico and another on the Caribbean side here. And again, we sequenced DNA from these different individuals, but different from what we saw with the red poles, we actually saw differences. So the fact that these bars are a different color implies that they're a different genetic cluster, a different genetic unit from the other ones. And so we found that there's this pretty deep split between the Western ones and the Eastern ones. And we actually were able to then compare this to all of the other species that were in the genus. And so when we did that, we actually found that these are not even each other's closest relatives. They're actually very distantly related to each other. So hopefully you have a little bit of understanding of how to read what are called phylogenies, which are these diagrams here. But the fact that the red lineage, the Western lineage, shows up here, that means that it's more closely related to Spirophila intermedia, which is a whole different species than what we thought its conspecific population was. So the collared seed eaters that were on the Caribbean slope, they're more closely related to these other species than they are to the ones that are on the west. So this would be like if we had humans and gorillas classified as a single species, but then we sequenced their DNA and we actually found out, wait, humans are more closely related to bonobos than they are to gorillas. And so that's kind of an analogy that we have here. And so this changes understanding of the biogeography of the group too. So now we have these 2 species that are distributed north here in Mexico, and a lot of these ones, the core of these Spirophila species are actually down here in South America. And so that suggests that there's been multiple colonization events out of South America, across Central America into Mexico. And so that kind of changes understanding of the biogeography and the evolutionary history of this group. So one sort of aside that I'd like to just briefly touch on is a project that I'm really excited about that I'm just starting, and that's a grant that I'm writing to focus on what are called Polylip Sky Islands. So this is a really, really fascinating ecosystem that occurs in the Andes Mountains of South America, and they're called Sky Islands because they occur at really, really high elevations, upwards of 3,500 meters, and so they're actually the highest forests in the world. And the way that they exist are they're sort of patches of this paramo or puna grassland, and then there's little strands of these Polylepis. They're kind of scraggly little bushes. You can see some of them here in this image. They're kind of stunted, scraggly trees that occur as forests and are islands of forests. And this is a really endangered ecosystem due to habitat loss, and so it's used for lumber by various people, and it's also threatened by climate change. But there's this really fascinating bird community, and so there's a lot of birds that specialize on these forests. And this presents an interesting question if we think about the community of Polylip Sky Island birds as a system, and we can compare across different species that have somewhat similar distributions. And we can ask, well, are they similar in terms of their differences in DNA? Or are there differences in terms of the divergence between them that might relate to some aspect of their ecology, perhaps dispersal, or perhaps how specialized they are on the Polylepis habitat? So this is something that I'm really excited to work on in the near future that will involve doing some field work at high elevations down in Peru and Bolivia. Okay, so I'm going to talk a little bit about some of the work I've done on birdsong, and for this I'm going to switch gears a little bit and talk about tanagers. And so I referenced tanagers in my last slide, but tanagers are this really fascinating group of birds. They're a single family, and so they're all related to each other, about 370 species and more, right? So that Intitanger was a new species that was just discovered and described, and so that number has been increasing in recent years. But they're this fascinating, extremely diverse group of birds in essentially any aspect of their biology. And so with color, morphology, song, you name it. They have differences that are pretty incredible. And so this was sort of the group of birds that I came up on and sort of learned how to do systematics and ornithology on as a master's student. And so this is a group that I'm continuing to work on. And as a master's student, I collected a lot of data related to their vocalizations. And so here are what are called spectrograms. And so these are visual representations of songs where you have frequency on the y-axis and time on the x-axis. And you can see that they differ among them. And it was sort of not fully appreciated before we had a good phylogeny or good understanding of the evolutionary relationships of tanagers, how diverse their vocalizations really were. But now with this new phylogeny, we have a much better understanding of the diversity of this group, both in terms of the color and other aspects, but in terms of their song too. And so they differ a lot in terms of how complex their song is, what frequency they are, what tempo the song is. So as a master's student, I collected a lot of data where I drew little boxes around notes in a computer program and analyzed thousands of songs and got a bunch of quantitative data related to the pitch, related to the tempo, related to how rapidly frequency is changing in the song. Basically anything I could think of, I was measuring. And so one of the questions that I then was able to ask with that is, do birds in different habitats tend to sing different songs? And this is something that's sometimes referred to as the acoustic adaptation hypothesis. It's the idea that vocalizations are a signal that's transmitted through the environment, and environments differ such that certain signals will transmit better through different environments. And so if you have a densely forested habitat, a lower frequency pitch that has a longer wavelength will be better to transmit through the forest versus a higher pitched vocalization. And so we were actually able to test this because we had variation in vocalizations as well as differences in habitat. And so we can combine those variables to try to test if there's support for this idea or not. And science is not always clean. We actually found differences in some of the characters, but not others. And so things like how rapid frequency was changing did seem to differ, but the actual base frequency itself did not follow these expectations. So the approach that I took with this project was across 300 species or more of tanagers. And so that's sort of a broad generalization. And I think that can provide some information, but also I think that there's a lot of times where if you were to drill deeper into those specific species, you could find actually instances that support this idea. So I'm continuing to work on song in different ways. And one of the things I'm interested in is repertoires. And so I'm continuing to focus on song as well, but sort of going in different directions and looking at it more on a population scale now. For instance, in the fox sparrow. I unfortunately don't have time to talk about that, but if you're curious, I can tell you more about that later. Okay, briefly, I want to talk about color. So color is also something I'm interested in. And for that, I'm going to talk about horned larps. So horned larps is another system that I've spent a lot of time thinking about and working on. They're a pretty amazing bird. They're a little brown bird, and sometimes I think they get overlooked for how interesting they are in terms of their biology. And so they have this incredibly widespread distribution. You can see that here. They're whole Arctic, and so they're in North America, but they're also in Eurasia and even in Northern Africa. And so they occupy a ton of different habitat types, from tundras to deserts to grasslands. And I'm interested in trying to understand how they're able to exist across all these different habitats, and how does this variation in the environment affect their coloration. So if you've seen horned larks in the wild, you might be used to seeing something like this, right? And believe it or not, there's a horned lark out there. But they're very well camouflaged, and they're very hard to see sometimes. These are some of the field sites that I've worked at. And you can see that there's some similarities in these field sites. They tend to be open fields, dirt fields, parking lot birds, they're sometimes called. I sometimes get jealous of other folks' field sites, but these are all different habitats from each other. And it's one of the things I was interested in is trying to quantify how the differences in habitat are impacting variation in color. And so when I was a postdoc at Berkeley, they had this amazing museum there, and this illustrates one of the nice benefits of museums. We have this incredible collection of birds across the Western United States, and so we can actually measure color, the dorsal color, so we can take photographs like in the top right here, we can take a photograph and we can measure color as well as patterning, so we can get an estimate of how much patterning there's on the back. whether it's uniformly patterned or whether there's lots of dark spots on there. And we can also compare that to data sets that we have on soil throughout the western US. There's been extensive soil surveys, and since larks are terrestrial, we can see whether or not there's a comparison there, both in terms of the color of the soil, the amount of carbon or calcium carbonate, but also in the composition of the soil, like how much silt versus how much sand and how much clay is in the soil. And, you know, as one might expect, we see pretty strong associations between coloration and larks. And so plumage and soil brightness are correlated with each other. So brighter birds occur on brighter soil. So in sandier deserts, you tend to have lighter larks. And also in terms of redness. And so in the Sacramento Valley, there's a subspecies that occurs in very clay-rich soil, and those birds tend to be very red in hue. And there's also differences in patterning. And so birds that occur in more granular soil, like in deserts where there's lots of sand, they tend to have more patterning matching their background. And so while this has sort of been thought to exist, this is sort of one of the first times we can actually really quantify across sort of a continental scale variation in pattern and color matching with these birds. All right, so I'm starting to run out of time, but I'm going to quickly talk a little bit about sort of an extension of the horned larks. And so with the horned larks, we might think about like what happens if the environment changes over time. And I was interested in this question, and to do that, I was able to actually leverage a museum series of larks from this region of Southern California. And so this is the Salton Sea, or near the Salton Sea, the Imperial Valley. You may know the Salton Sea. It's a pretty big birding destination. There's a lot of migratory birds that come through there. And so it's a pretty weird place. It was created by an accident from the All-American Canal. It created this man-made lake. And so we have this desert ecosystem within which there's this huge influx of water and agriculture. And so I was curious, what happens to the birds that live there and that persist? And so larks nested in the desert flats and the mesquite flats before the rise of agriculture in this area and are still there today. And we actually were able to compare larks that we collected recently to larks that were collected from the early 1900s. And so this series here is a series of birds that were collected by ornithologists in the early 1900s. We went back and collected some, and you can see it's a little bit hard to tell, but they are darker. And this definitely comes through when we're quantifying the plumage. And so it seems like the birds have gotten darker over time. And they've also actually changed their diet. I won't be able to get into that either. But we've been able to use this series of museum specimens to look at how larks as a species have changed over time in response to the rise of agriculture in this region. So yeah, so as contemporary larks have persisted in the area, they've gotten darker. They actually occupy a lower trophic level, so they seem to be eating more seeds and less insects than they did in the past. And they're also actually incorporating water from a foreign source. And so we can figure all this information out because we have specimens from the past. And that's going to segue into my last bit, which is thinking about Louisiana avifauna and one of the things I'd like to do while I'm here in Louisiana. And so one of the things that I really believe in terms of the value of natural history collections is emphasized really well by Joseph Grinnell, and he was the founder of the Museum of Orthology at Berkeley. But he says the greatest value of the museum will not be realized until many years, and that the student of the future will have access to the original record of faunal conditions. Right, and so, you know, the ornithologists in the early 1900s collecting larks out in the middle of the desert had no idea that I would use those as a comparison point. But I think that's a really good example of why we need to continue collecting specimens so that generations from now, people can use the specimens that we collect today as a baseline, as a reference point. And so we can think about the LSU Museum of Natural Science as transecting the environment and changes in phenotype or the appearance of birds, as well as the genetic composition of those populations through over a century of change. And if we want to care about how things change in the future, we need to have continued collecting in order to do that. And so one of the things that I'm hoping to do is to improve our collections of Louisiana birds. And so this is just, again, a heat plot showing where we have the most specimens. So here we are in Baton Rouge. We have a lot of specimens from Baton Rouge. We also have a lot of specimens from southwestern Louisiana, all along the coast, but relatively fewer specimens from northern Louisiana. And also, I should mention that this is across all seasons. So most of these birds were collected during the non-breeding season. So if I were to limit this to the breeding season, which I should do, we have far fewer. And so there's actually quite a bit of work to do locally to try to document our biodiversity. And one of the, this would allow us to track changes in phenotypes and diet, and also things like exposure to pollutants, like pesticides and other chemicals that enter the environment. And just as an aside, really briefly, one interesting thing that happened is, if you remember the crazy snowstorm that we had, I had just moved here. I was like, oh, it doesn't snow in Louisiana. And then we had that crazy snowstorm during Mardi Gras. And you might have heard, I think, I can't remember what her name was, but I think someone actually gave a talk to y'all about the purple martins. And so we actually have a lot of specimens from Texas and from Louisiana of purple martins. And we're collaborating with some of the other folks involved in that project, someone from South Carolina, University of South Carolina, to try to understand, what happened to these birds physiologically. And so, as an example, we need purple martins from the past or purple martins from the future as comparison points. If we only had purple martins from that snowstorm, we can only understand so much. And so this sort of speaks again to the importance of general collecting and sort of the role of the museum as a way to try to understand changes in biodiversity in populations. All right, so I just want to end thinking a little bit about the future of the museum. And so one of the things that I think is really important to think about in museums and STEM more broadly is diversity and inclusion. And so you probably noticed when I spoke about the history of the museum, it's a pretty homogenous group. There's a lot of white men in the museum, but fortunately this is changing, and it's something that we're dedicated to here at the museum. So one of the really awesome examples of this is this group of women that led the Emily Sneffledge expedition. So Emily Sneffledge was a really accomplished ornithologist in Brazil. And so a group of LSU affiliated women led, I think, the first all-women collecting expedition to Brazil. And so they were on the Amazon, living on this boat, collecting birds, and in a region that really hadn't been studied before. And so we're looking for ways to try to improve diversity within the museum, and we think that's an important thing to do. Another thing I just want to speak to is one of the constant things that's difficult in a museum is both space and funding. So it's interesting reading that first thing I linked. You can hear George Lowry and how concerned he is about space, and we've been constantly stretched for space. Some of our cabinets are overflowing. So this is just a quote from that. And this is something, an issue that's been happening since the 1930s. And if you've ever been to Foster Hall, which is where our museum is, you know that it's split between art and the Natural History Museum. And so the art component is on the second floor, and there's painting studios up there, and then the museum's on the first floor. And we've been struggling with the university to try to gain access to the rest of the building, which was originally promised to George Lowry back in the 1930s. And it's been this ongoing struggle for decades and decades and decades. But just recently, we've actually gotten permission to the rest of the building. So there's going to be a new studio art building on the south side of campus. And in 2023, and I think onwards, we will have access to the rest of Foster Hall. And this is a huge, huge relief for us curators. And George Lowry would be thrilled if he were to hear this. He would be ecstatic. So this is really, really great news for us. It's sort of a time of change in the museum. I'm a new hire. We have Steve and Donna who will be retiring soon. And, we have a new search for a genetics curator going on right now. And so it's sort of a new chapter in the museum, both in terms of personnel, but also in terms of space. And so it's an exciting chapter, but there'll be a lot of hard work to do to make sure that it gets done properly. And so I just wanted to end by saying that there's various ways that you can support the museum. We would love any support that you can offer us. We have a lot of different social media pages. I'm going to put those in the chat right now. And we have various fundraising events that we do too. Let's see, I think that worked, yeah. So we're active on social media. We like to celebrate our successes and share our activities through that. So I encourage you to check out those pages. We have a lot of outreach events for all ages. So we have stuff that are geared towards school kids, that are more geared towards older folks as well. So across all ages, we have various events. We have an annual Big Day that's geared to support student research. And then we're probably going to have a lot of fundraising opportunities coming up to try to help in our move to the rest of Foster Hall. But really, what I'm hoping is that we can enjoy birds. Really, we're all naturalists. We do this because we love nature and we love trying to understand nature. And enjoying birds together and conserving and trying to understand nature is our main mission and main prerogative. But yeah, so with that, I think I'll end because I'm running out of time. But thanks for your patience, and thank you very much for listening. I just wanted to thank some of the folks that contributed talks and some of my funding sources, and thank you.
Thank you so much. You covered an incredible amount, but it wasn't overwhelming. It's just terribly impressive. We're not going to get kicked out of the Zoom right at 8. I do not believe. I didn't set this one up, but I don't think it should be set up to end this call. So if anybody has any questions, by all means, put them in the chat or unmute yourself and your video if you would like, and kick any questions towards Nick. I'll pause and give everybody a chance to.
Yeah, I'll also just say while folks are formulating the questions that, once we unfortunately don't really have the museum open right now because of Omicron and because of COVID, but hopefully in the future, if you would like to come see the museum and also see behind the scenes research part of it, I'd be happy to coordinate a tour with people through Audubon anytime.
I have a question.
Yeah.
Yeah. So I was really intrigued by the correlations between the different soil types and the patterning and colors of the birds.
Yeah.
But those are just, correlations.
Right.
It doesn't really establish cause and effect. So I'm curious as to what steps you could take to do that.
Yeah, that's a good question. I think, it's tough. You could try to do some kind of experimental setup potentially where, I don't know, like if you had captive populations of larps, I could imagine you could, interbreed them and see potentially if you have populations on different soil types in little mesocosms or something, you could do something like that potentially to try to get at, is it selection that's really driving this? But yeah, getting at that causation is tricky, and it's something difficult that we face working with birds and non-model organisms in general, right? We can't have controlled experiments that we need to really eliminate those other possibilities. So yeah, I mean, it's tricky. It's something that's come up before, and I think without having actual captive populations, it's difficult to say it is the soil type and not something else that is immediately affecting this variation.
Well, has that been shown with other bird species where you see these relationships between soil types and coloring or patterns or?
Not that I know of in birds. There's with mice, so Microtus mice. So Hopi Hoekstra has worked on a mice system, and it's analogous to the horned larks, where there's sort of dark mice and there's light mice. And she had basically like these enclosures where they would release mice, and then like over time they would go back and see which mice survived, and they would find in the different soil types different ratios of survival. So they were able to demonstrate pretty conclusively that it was related to soil type, but as far as I know, I'm not aware of any analogous studies in birds. Yeah.
Oh, it's fascinating. Anyway.
Thank you.
So the horned larks, what kind of predators do they have to worry about?
Yeah, so mostly
you would think the difference in soil type and whether they can be hidden or not would be impacted. That would impact if a predator was after them.
Right. Yeah. Good point. So yeah, they're mostly worried about like hawks, accipiters, raptors. So visually oriented birds from above is kind of the way I think about the predator visual model. So yeah, but
I'm picturing a mock up of the environment with like a dummy bird there and have a hawk. If you have a trained hawk, you know, that might go after it type of.
That could work. Yeah, you could if you had like you mean like if you had like a model, like different model larks that you put out, you could see which ones the hawk strikes at. Is that what you're thinking?
Yeah.
Yeah, that would be great. That would be super cool. Yeah, I need to work on my talents to make a mock lark, but yeah, that would be neat. I think that people have done similar things like that with I can't remember. I think it was like caterpillars or something, but we're not. Well, with bird models too. I think I can't remember a study. I can't remember the details of it, but yeah, that's certainly one way you could go about it for sure.
Nick, sticking with the larks, for the ones that showed the plumage coloration pattern over time, the Salton Sea area, how long was that time span?
Yeah, so I think the All-American Canal was constructed in like 1905, something like that, and then I think it burst in the teens or so. So the birds that we collected were like from 1910 or so. Sorry, the birds that we compared it to, the historical birds, were from 1910. And then the contemporary birds were from like 2010, 2014 or so. So roughly a century or roughly.
Interesting. Colette was asking if the museum's going to collect more birds throughout the state from window strikes.
Yeah, I think that's proven really effective for other museums. So the Field Museum in particular in Chicago, well, unfortunately, they have a lot of window strikes that come through, especially in migration, but they have a very active group of people that survey the large buildings downtown and look for birds that have unfortunately hit windows. So I think that's something that we could do in downtown Baton Rouge, and I think it'd be great to do that. And we certainly take salvage birds. So if anyone finds a salvage bird, and it's in good condition, we would love to take it in and hopefully make a specimen out of it. So that's definitely part of the way that we are hoping to kind of increase coverage and representation is through sort of public salvage. Yeah.
For the public salvage, do you have tips on that? Do you want people to freeze them, label them a specific way?
Yeah, thank you. So yeah, the way to do it would be to essentially, if you can write down the date, where you found it as specifically as you can, your contact info, and then put it in your freezer. It might seem weird to put it next to your frozen veggies or whatever, but it's harmless. And then whenever you can, you can contact myself or Steve Cardiff, our collections manager. And if you're in town, you can drop it off at the museum, just at our front office, but basically getting it onto ice as quickly as possible helps to preserve the DNA and also prevent sort of degradation of the carcass.
All right, so one more question in the chat is about the tours that you mentioned. And when you start them back up again, if you can let our group know. And I don't know how you guys proceed with that, because at least for the behind the scenes tours, I know you try to keep those groups pretty small.
Yeah, for that, it would probably, it would depend on the group size, but, typically we have, I don't know, anywhere from we could do a group of 4 to 8, but if we had more than that, we could split it up because the museum is pretty large, and so we could have one group starting in one part of the museum and another starting in another, and we can do that. But yeah, we can definitely keep in touch. I'll let y'all know. Hopefully, I'm optimistic that, it's impossible to know what's going to happen with the pandemic, but I'm optimistic that maybe, by the end of the spring semester or in the fall, certainly we could open up and have behind the scene tours again. Hopefully soon, fingers crossed.
Yeah, I guess that applies to the Master Naturalist group also, for anybody who hasn't taken those courses, which Colette helps to organize the birds section. They normally get to do the tour as well, but I guess we'll see what happens in the spring.
Yeah, we can do smaller groups, but yeah, I think for now we're just kind of holding tight and hoping that things improve.
Well, thank you so much. I don't
Yeah.
want you to wait into your night either, without hearing anybody hype up in the next few seconds or seeing any additional questions in the chat. I can close it down, but it's fascinating, and congratulations on the expansion and the new rooms that you get to move into, because I was certainly curious right before you even said that, how you were even going to continue to collect at this stage.
Yeah, it's a huge relief. We're thrilled about it. It's going to be an exciting time, definitely.
So back in the day when I was an undergrad, they used to boast about LSU's collection being like of the top 3 in the world. I don't know if we still do a ranking system like that, and it shouldn't matter, but it's still just kind of fun to know where we rank.
Yeah, we're still number 3 as far as I know. We were 4 for a while, but I think we passed Kansas, and now I think Harvard is still in front of us, and I should know this, but I can't remember what the other one was. I can find out and stuff.
I didn't mean to put you on the spot with that one.
No, it's all good.
I was always taught that that collection put LSU on the map before football did.
That's good. I like that.
Katie, he needs to keep us because we need to know how to brag. You've got to give us all the deets so we can brag. We love doing it.
Yeah, totally. So yeah, it's the 3rd largest behind Harvard, and Michigan was the other one that I forgot about. So University of Michigan is the other one that's ahead of it. 3rd largest university-based collection in the world, and the largest collection of genetic resources in the world. So that's
number
So
one.
what kind of numbers are we, when you say that, what kind of numbers are we talking about?
For the genomic resources?
Well, the collection, based on the collection. So what kind of, if you're comparing us to Michigan and Harvard, what's the difference in numbers?
I mean, I don't know off the top of my head, to be honest. And there's also, that's university affiliated. So there's also collections like the Smithsonian National Museum that are very large, and the American Museum that are very large too, larger than ours. So in terms of university affiliated collections, we're the 3rd in the world. But then, so I mean, our numbers are like 220,000 is our current count. But then for genetic resources across everything, it's 140,000 individuals of about 8,000 species. And I think that's across all vertebrate groups. So, but I think part of that is related to us being a relatively young museum compared to some of the other university affiliated museums and other museums that are out there. Since we were founded in 1936, most of those other ones were established, late 1800s, and that was when a lot of scientific collecting happened, was sort of late 1800s, early 1900s too. So the fact that we're as high as we are is a testament to how active we are too.
Can I ask another question? Just because I'm reading the Feather Thief right now.
Oh yeah, it's a great book. Yeah.
And so they talk about skins.
Yeah.
Can you explain? Because I'm not sure. I mean, when I think of skins, I think of like feathers on top of the body of the bird. So what do they mean by skin exactly?
Well, I mean, it's basically like a museum specimen. When they're prepared for a museum, they're compact and they're on their back, so that they fit many into a single drawer. Yeah. You can still study their phenotypes and what they look like. That's like, as opposed to like a mount, right? So this is like a fulvous duct. That's like a mount. So a skin is like sort of the classic representation. I can find a picture for you. Hold on.
Maybe others already know that, but I don't.
No, it's a good question.
I'm really relatively new to this.
I'll share my screen again. So I don't know if you can see that, but those are like study skins. So like this kind of how it's the wings are folded in, it's laid on its back.
Huh.
That's what we call a study skin, as
Oh,
opposed,
okay.
yeah. And so
So
the
it's
feather,
so basically it's just the posture of the whole bird in a certain position.
yeah, yeah, as opposed to, so sometimes we have skeletons where the feathers aren't attached to it anymore, or we have specimens that'll be in alcohol, so it's the whole bird, including the internal organs that are preserved. With the study skins, most of the viscera, most of the internal organs are actually removed. So that's what we call it a skin, because it really is just the skin, essentially, a part of the bill, part of the wings, and part of the legs, and then cotton, and then it's sewed up. So, yeah.
And the feathers don't lose their hue?
No, they stayed pretty good over time. There's if that's partially why we have them in the cabinets, is to prevent the UV damage from happening.
Mhm.
Sometimes there's what's called foxing, which is like where there's changes to the color over time. But typically, they stay in really good condition, and you can study specimens that are 100 years old and still get a pretty good assessment of the color and the patterning of the plumage.
Thank you.
Yeah, my pleasure. Well, thank you all for listening. Appreciate it. And yeah, hope you all have a good night. Feel free to get in touch anytime.
All right, thank you. We will. Have a good night, everybody.
See ya.